So the axon eventually becomes one or more axon terminals. The axon terminals are regions in the neuron responsible for signal output into the synapse. So this actually brings us full circle back to the beginning of the unit, because here once again we're talking about an axon terminal and a synapse. So the only difference between right now and before is that this neuron that we've been talking about is actually the presynaptic neuron now, because it's the one that's sending a message to the next cell. And it's doing that by releasing neurotransmitters that are stored within membrane-bound vesicles inside the axon terminal. Now, most axon terminals tend to have this neurotransmitter made and ready to go. And so all they're really waiting for is some kind of release signal. And that signal is calcium, which is an ion that we haven't talked too much about yet. But calcium allows the vesicles to move to the presynaptic membrane and attach or dock to a family of proteins at the membrane called snares. Those vesicles can then exocytose the neurotransmitter into the synapse, and ligand-gated channels on the postsynaptic dendrites can start this entire process all over again. So the only thing that we really have left to do in this unit is to establish the relationship between the action potential, which is due to sodium and potassium, and calcium, which we need to release neurotransmitter. So this picture is trying to represent the channel composition of the axon and the axon terminal. All along the axon, you can see the voltage-gated sodium and voltage-gated potassium channels in each segment. But when we get down to the axon terminal, you'll notice that we really don't see those anymore. Instead, we start to see voltage-gated calcium channels, which I've represented here in black instead. So as the depolarizing sodium spreads from the last part of the axon segment into the axon terminal, it brings the voltage-gated calcium channels to threshold. That allows them to open. Now, remember from before that calcium is at a relatively higher concentration outside the cell and a relatively lower concentration inside the cytosol. So when those calcium channels open, it allows calcium to enter the axon terminal and allows those vesicles to dock and to release neurotransmitter. So now that we've talked about this whole story, one of the things that you want to think about is how changing the concentrations of ions like sodium and potassium and calcium inside or outside the cell would interfere with the action potential or neurotransmitter release, or both or neither. Alternatively, you could also think about how a drug like tetrodotoxin, which inhibits voltage-gated sodium channels, would impact this entire process. So, summarizing everything that we've talked about from this unit, we talked about how action potentials travel down the axon, down the presynaptic axon into the axon terminals, and bring in calcium so that neurotransmitter can be released into the synapse. The neurotransmitter can then cross the synapse and bind to ligand-gated ion channels on the postsynaptic dendrites, and that starts a graded potential that will eventually initiate the action potential inside of the postsynaptic cell. That action potential will then spread down to the axon terminal, trigger the release of calcium, trigger the release of neurotransmitter, and start this entire process over again a second time inside of a new postsynaptic cell. So this process can continue for as many cells as we have in the chain. In most cases that we'll talk about later, there's going to be at least 3 neurons responsible for sending any particular sensory or motor signal inside the body.